Okay, let's talk about chapter 1. I have cut the number of slides tremendously. If you did watch the first PowerPoint, that was my very first recording. So it was kind of like just a trial and error kind of a thing. But I think I'm getting better at this. Okay, so let's go back over chapter 1 in more brevity. All right, so when we start talking about biology, we have to remember that biology is the study of life. And when you ask a question about life from a scientific perspective, then you are in effect like doing biology. So one concept that you'll keep seeing pop up over and over again is evolution. So there's a formal definition of evolution on this slide. It's right here, very last bullet. But speaking, I guess, like a little more loosely than how this is stated, when we think about evolution, we have to think about it as a gradual change that happens over time for a population of organisms. And what you'll see is that depending on different things that happen in the environment, there are certain organisms within that population that may be able to better adapt because of some characteristics that they have that others in their population may not have. And I'll give you a specific example with these mice that they speak of in the textbook. So evolution is going to be the thing that drives and shapes like the differences that we see on this planet currently. So here are the two mice that I was talking about. They're both the same exact mouse species. That just means that if these two mice were to come in contact with each other, they could mate and successfully reproduce a brand new mouse of the same type. So why do they look so different? The reasoning is because of where they live. So the one that's lighter colored lives in a beach area, so that's the beach mouse. And then you have one that lives more inland, but they're not far from each other. Like they could very easily cross paths. Their coat color differences are important though for their survival depending on which environment they live in. So we'll revisit these mice towards the end of this. All right, this I've already said, biology is the scientific study of life. So biologists can ask really big questions or they can ask very small scale questions. Big scale questions tend to deal more with like a bunch of organisms, how they interact with each other, that type of thing. And then the small scale ones could be something as simple as what gene is responsible for creating whatever characteristic you're interested in observing. So I call that small scale because you're looking at DNA when you're dealing with that. Large scale is more like a bunch of organisms together interacting with each other. All right, so the first concept in this chapter is the idea that things will be spoken about or written about in terms of different themes. So there are 5 main themes. They call these unifying themes throughout the book. So organization, information, energy and matter, interactions, and evolution. Let's go over those quickly. So in the first theme, we're going to talk about biological organization. All right. So life can be looked at from different levels. So the idea is you can look at life as just a single cell, or you could look at life as like the bigger picture, every living thing on the planet. So here's a picture of the planet that we live on, planet Earth, and they call this the biosphere. So the biosphere is going to include every single region on the Earth where you can find life, and that is everywhere. There's microscopic life, meaning you can't see it just with your naked eye. You need to be able to look under a microscope or magnify in some kind of a way. And what we're getting ready to do now is this idea called reductionism. So we're going to start with the whole globe, and we're going to scale back until we can't go back any further. All right, so here's our first scaling back, or our first reduction that we're going to do. So we went from the whole planet just now. Now we're speaking specifically about an ecosystem. So an ecosystem is something that's going to include living and non-living things in a specific area. Ecosystems have some characteristics that they share. In other words, like you could have a desert ecosystem. So in those places, of course, you would expect to see a lot of sand. It would be very hot in the day, very cold at night. Organisms that live there, they should be having some characteristics in common with each other, like their ability to survive has to be suited for that particular ecosystem. You could also look at something like the jungle or the frozen tundras, or the list can go on and on. All right, so now let's scale back again. So we're going to reduce another level. Within the ecosystem, you have these things called communities. And a community is not considering the abiotic or non-living things. Now we're just looking at the living things within the ecosystem or within a small portion of the ecosystem. So in this picture, they're showing that there are plants in this community, there are animals in this community, and again, they're things that we cannot see with our naked eye, part of this community. So now we're going to pull back another layer. We're going to reduce even further. So instead of looking at the deer and the plants, now we are looking at one specific population within that community. So in this case, we're looking at these purple flowers, or they're like a bluish purple flower. So a population is going to be a group of organisms that are the same species within a community. All right, let's scale back again. So now instead of looking at all of those plants or all of those flowers, we're just going to select one single individual from that population. So this is what we call an organism. It's just the one specimen by itself. So let's scale back again. All right, now we're looking at the parts of that organism. Notice that we're getting smaller and smaller every time I click to the next slide. So they are listed here. They're saying organs is the next level back. And they use a plant as an example, but you could use anything else like a human or a deer or, I don't know, fungus, whatever you want to put. These things can always be scaled back. All right, let's go in a level deeper. So now we're peeling back even more. So we're not looking at the organs, we're looking at the tissues that make up those organs. So depending on what organ you're talking about, you get different types of tissues. And if we scale back or reduce again, we get to cells. So cells are the things that make up the tissue. This is where you would need a microscope most of the time to be able to see what the tissue is made of. So in this case, we're looking at a plant cell. It looks green on the inside. Each of those little teeny globular looking things that are within that white outline, they have a name. If we scale back further, this is what we're talking about. We're talking about something called an organelle. So an organelle is nothing more than a teeny tiny part inside of a cell that has a job to do. Scale back one more level. So what are organelles made of? Organelles are made of molecules. And from there, we could scale back one more, and we could go so far as to say every molecule is made from atoms. So for the sake of this class, we will talk about, or we have talked about, molecules already and atoms. But when we talk about what is the first thing that we consider actual life, like what is a living thing, we're talking about the cell. So the cell will be the smallest unit of life for this particular class. So here is an overall view of every single part that I just went through. You start with the earth, then you scale back, and now you're looking at a specific part of the earth. That's an ecosystem. Within the ecosystem, there are communities, and within the communities, there are populations. If you were to select one organism from the population, you could look back further, not look back, but scale smaller, looking at its individual parts. From there, you could look at the parts that make up the tissue, which would be the cells. The cells themselves are made of organelles, and the organelles are made out of molecules, which are made out of atoms. And again, this whole thing is called reductionism. All right, so as we move from something that is smaller up to the larger scale thing that we're going to look at, I mean, it depends on what kind of system we're talking about, but let's just keep it very general right now. What you'll find is that there are some properties that will begin to emerge once you kind of zoom out. So like an atom by itself may not have too many interesting properties going on, but once you combine a few of them, so a few different kinds of atoms together, you get a molecule. And molecule behavior could be very different from the individual atoms themselves. And then if you were to put those molecules all together, the behavior of the molecules as a whole could be very different from just if it was a singular molecule. So what you should start to see is the more and more you zoom out, the more and more properties or characteristics may show themselves or present themselves to you. And they call this in the book emergent properties. So this is one idea that you're going to see several times over. The book gives an example of like a bike, I believe. So they say like if you go in a room and you notice that there's like nuts and bolts on the floor, and maybe you have like a frame and a seat, some other things, and wheels. That doesn't make that thing a bike. It's the way that you put it together. Once you put it together the proper way, then you get this new emergent property. You're able to actually get on that item and ride, and it can take you somewhere. All right, so I just alluded to this in the last slide. Systems biology is nothing more than observing smaller parts of a whole. That's all you're doing. And the examples for using systems biology, they can range from something macro. So you could be looking at the ecosystems within the biosphere. That could be like a form of macro systems biology. Or they can be very micro. So you could look at the organelles within a cell that make the cell able to do whatever job that it needs to do. Again, that's still systems biology. It's just on a very smaller scale. All right, this slide is speaking on how structure and function can be discerned from you looking at the parts. So it says at each level of the biological hierarchy. By the way, biological hierarchy is nothing more than that whole thing that we just did where we were looking at the biosphere and then we reduced all the way back to a molecule. So at each level of the biological hierarchy, we find a correlation between structure and function. And this is important because if we run across something that we've never seen before, what a scientist would be most likely to do is to study whatever it is, and then they want to try to find something that we currently know about, and they may compare the two. And sometimes you can kind of guess, not guess, but like predict, I guess is a better word, what the behavior of this thing that you found will be. And I'm being very vague right now, and that is intentional. Just keep in mind that there is a way to determine structure and function. I'm looking at like my shoes in my room right now, and I just thought of an idea. So if you guys owned, like, let's say we all own only one pair of shoes, and there's some specific things about shoes that make them shoes. So if I say, okay, well, it has a sole that I can put my foot like on, right? And then maybe there's some material around the sole that kind of covers my foot, and it has shoelaces. And if that's all that I know about the shoe, if I were to go out in the world and let's say, I don't know, I'm walking down University Boulevard and I come across a shoe, maybe I don't know it's a shoe because I only have, we all only know of one type of shoe. So let's just say maybe that the shape of this particular shoe is different. If I were to pick it up and inspect it and I notice, oh, it has a sole, oh, it has like a covering, oh, there's some shoelaces, then I could infer that it's a shoe. And I know that sounds so stupid, but like that's basically what it is that scientists are doing. They're taking something they know about, and then they compare the thing that they do not know to the thing they know, and then you come up with some properties or characteristics of this thing that most likely will, come to be true. Okay, so I've already told you this. The cell is going to be the most basic unit of structure. So once we get all the way down to a cell, when we look at the inner workings of a cell, the organelles themselves are not life. The atoms that make them are not life. The molecules are not life. The cell is going to be the most basic unit that we consider as being like living. So there's two types of cells that we're going to come across in this class. Both are listed here in the third bullet point. So they're prokaryotic cells and then they're eukaryotic cells. So what is the difference? All right, eukaryotic cells are going to have two things happening inside of them. Not happening, but they have two things present inside of them. So eukaryotic cell is going to have organelles and it will also have a nucleus. That's it. Prokaryotic cells, they're not going to have a nucleus and they will not generally have organelles. Here's a picture of two of them side by side. This is just for scaling effects, so you can see how much bigger a eukaryotic cell is when you compare it to a prokaryotic cell. So here is the eukaryotic cell, so all of this area here, that's the cell. Prokaryotic cell right next to it, very tiny. So prokaryotes are usually bacteria. Eukaryotic cells are animals, plants, fungi, things like that. So both of those were just the same picture, just separately. All right, so here's the next theme. How do we pass on information? We pass on information through something called DNA. So DNA contains genes, and we pass the genes on from one generation to the next via reproduction. In this case, we're looking at some reproduction that's happening. So you have like one cell they're showing you here. So this picture here is one cell. And what you'll notice is in the center, so in this region here, there's a bunch of like blue stringy lines. Those blue stringy lines are the DNA. Sorry. Those blue stringy lines are the DNA. And the whole thing that I circle is the cell. But this particular cell is doing something very important. It is actually splitting from 1 to 2. So it is making a copy of itself. It is reproducing. And then, so here in the second picture, you've got, it would be one cell here and then one cell here once it's all done. All right. So genes, again, are nothing more. They're segments of the DNA. That's all they are. Just pieces of the DNA that code to make specific proteins, or they code for like specific characteristics that you may see in another individual or in a plant, or whatever living thing we're speaking about. All right, so humans, this is how we reproduce. It's not just us. There's a bunch of other species that reproduce in the same way, but basically you have an egg cell. This usually comes from a female. Not usually. It comes from a female. I don't know why I said that. And then you have a sperm cell, which will come from a male. And what's interesting about this, or these particular sex cells, is that they contain half of the amount of the genetic information you need to make a whole new being. So that's why they say like, oh, you're 50% your mom and you're 50% your dad, because your mom's egg cell gave up half the number of chromosomes they needed to make you, and then your father's sperm cell did the same thing. And so once it's fertilized, this is when you get the whole new being. From there, you have some division that happens. It's called mitosis. And don't let that word scare you. We will speak about it when we get further along into the book. But the first single cell that made you, it will divide rapidly. over and over again via mitosis, and then in the end you end up with a brand new baby. All right, let's talk about DNA as a molecule very, very briefly. So DNA, it is short for deoxyribose nucleic acid. The only things that you need to know about DNA right now is that there are molecules that make the DNA, and that the molecules that make it form this particular structure called a double helix. And a double helix looks like a ladder that's twisted. There are some specific building blocks of DNA, so things that make up DNA. But what you'll find is that the rungs on the double helix ladder, they're going to be one of 4 bases paired to each other. So you can have A and T as one ladder rung, and G and C as another. And the variation between like the combinations of the rung is what makes each one of us different, individually different. So maybe on my ladder rung, my first one would be like the AT, and my next one could be AT, and I could have 50 after that were all AT. Whereas maybe one of you has AT for the first rung, and then maybe GC for the next one, and AT for the rest of them for, I don't know, 100 base pairs long, or whatever you want to say. But the point here is that there's a lot of variation that happens just from the way that these molecules link up, and the way that they are ordered within your own strands of DNA. All right, here's a picture of it up close. So this is doing that reductionism thing again. Instead of looking at the biosphere, we're now just looking at a single cell. And then when we zoom in to the center, you're looking at the DNA. So here is the DNA made bigger. So they like blew it up for you so that you could see close what it looks like. And when I was talking about that whole ATGC, the way that things are ordered, and I was referring to them as ladder rungs, I'm talking about like these things. I'll do it in black so you can see it easy, like these. So these guys right here, some of them are harder for me to draw the line through because of the twisting that's happening. But then here, right next to it, they show you what it would look like if it was untwisted, and they drew this as a model so that you'll be able to understand this concept more concretely. It's not as abstract when you draw it like this. So that's a model. All right, so how do genes become expressed? That is a thing that is important because it's going to tie us to different genetic disorders, diseases, and then some things that are like a lot less ominous, just like what are your physical characteristics, the texture of your hair, the color of your eyes, so on and so forth. So gene expression is just when we take that DNA, and not we specifically like thinking about it, but like in your own body this happens. So your DNA is uncoded basically, and then after it's uncoded, it produces specific parts that will make you look like you, or make a disease come, or make a disease not come, so on and off. All right, so here's a picture of what this would look like. In this particular example they're giving you, it's very, very specific, and I think they did this intentionally so that you can understand how intricate this process is. But what you have on the left is someone's eye, and they are doing the reductionism thing once again. And they're saying like this gray picture here is just a bunch of the lens cells from your eye. And around the lens cell, you have these proteins that are called crystalline. So that's what you see in figure A. When you move to the other side of this figure B, they're showing you the process of taking your DNA, finding where the gene for crystalline is, unraveling it, decoding it, and then finally giving you a protein that would be found on the outside of each of these lens cells in your eye. So, I mean, like, that's crazy, crazy specific. And it seems like it really isn't that big of a deal, but it's amazing the amount of detail that goes into something that is so small. And if you think about all the other small, small things going on within you, it's amazing, like, that your body is doing all of this stuff and you're not putting any kind of brain power to it. It's just doing it. These molecules interacting, these atoms interacting, and then these cells interacting. So here it is zoomed in. And then there's the process, or half of it anyway. And then there's the end of it. So again, you started out with DNA, and then your genetic code gave some indication that it needed to make this particular protein, and so it would do that. And we'll talk more about these processes when we get into a further chapter. Okay, so let's move on. Genomics. Okay, my son is screaming. Hold on. Okay, I'm back. All right, so genomics. The only thing you need to know about this is that there have been a lot of people that have put their whole their hard work into this process of gathering all the possible information that they can about the genome of various species. And what is a genome? It's just a collection of the genes that a species has. So we, as scientists, we had the desire to catalog them, or to put them within specific boxes. And why would you want to do something like that? Why would you need to catalog? The reason is because we don't know, there's a potential that some species may be able to overcome maybe some of the illnesses that plague our specific species, or vice versa. We learn a lot about ourselves and about the world around us through viewing the genomes of various species. Underneath of that, you see the word proteomics. This is essentially the same idea as genomics. The difference is, instead of looking at an entire genome, you may be focusing on a specific protein. So think about that eye example that we just looked at. The gene for making crystalline, that would be the genomics portion of it. Finding the code that makes, or that is going to result in that particular protein being made. But proteomics would only study the properties of the crystalline protein. And I hope that makes sense. Okay, bioinformatics. Literally, there is so much data that for one human to have to process it all, I don't think it could be done in a lifetime. I think it would take several hundred lifetimes over in order for you to be able to do this by hand, like cataloging all this stuff, keeping the information, finding similarities. So naturally, we tapped into the tech potential that we have, so technology potential that we had, and there are a lot of great people who have come up with different softwares and ideas for how to store all of this information, and not only store it, but also like sift through the data. So that's what bioinformatics is. It's just like itemizing and then also analyzing all of this biological data that we've collected. All right, so here's the next theme, talking about transferring energy from one organism to another. This is how things are able to grow, develop, and then in effect, it affects everything else that goes with life. So reproduction, metabolism, etc. All right, so where is the main energy source coming from? Everybody should know this, and you need to remember this. It's all coming from the sun. I hope you like my second grade drawing of the sun. And then I'll just make him smile because he's giving all the energy, or it's giving all the energy. So the main input is coming from the sun. So maybe in elementary school, you heard a teacher say the word photosynthesis. And then maybe you heard it again in middle school, and now you're hearing it again in high school. And what you should know is that we're getting more and more specific about what exactly the process of photosynthesis is. So I'm going to give you just the very big picture of what photosynthesis is. The sun shines down on a plant, and then the plant uses the sun's energy to make sugar to keep the plant alive. So how do we transfer that energy? Well, we have to eat the plant, or something else has to eat the plant, and then we have to eat the thing that ate the plant. So you get the picture here. The initial source of energy is the sun, and then plants are the first ones to kind of convert that energy into a usable form down here. So when we talk about energy flow through an ecosystem, it comes in usually as light and heat. That's what the sun gives off. And then we recycle it via these chemical reactions, photosynthesis and cellular respiration. There's a whole chapter dedicated to that. So here's a picture of what we just said. Here's the light coming in. There's the plant converting it into a usable form. We have a caterpillar on here eating the plant, and something would eat the caterpillar, and then the thing that ate the caterpillar would eat maybe more caterpillars and store up a bunch of energy, and then something has to eat that thing, and so on and so forth. Yep. Okay, next theme. So organisms interacting with other organisms, very important. Interaction is key to survival. You cannot live by yourself. But we're not only talking about interactions of organism to organism. You could be talking about the interaction between something living and something non-living, like the sun and a plant. So let's keep going. Okay, this is just more about interactions. So interactions affect individual organisms and the way that populations evolve over time. That is completely true. You can think of I can think of a ton of examples to give, but it's not worth it here. Here's a picture of some interaction happening that it's beneficial to both of the organisms that you're looking at in this picture. So the yellow tangfish, which are the ones that are yellow, they look kind of like flatfish. And then you see like the sea turtle. And you notice that the sea turtle is not bothered by the tangfish. It looks like the tangfish are like eating something off of him. And in effect, that is what they're doing. They're clearing some debris, maybe like from his shell. And so they get a meal out of this deal. And the sea turtle gets a cleaning, and that cleaning helps him be able to glide better through the water, so everybody's happy and we can all go on with our day. All right. Climate change is something that is a result of human interaction, not just human interaction, but we have noticed that humans are starting to, speed up this idea of climate change by some of the things that we do. So our interaction with the environment around us could be potentially destroying the planet. So climate change, the definition, just in case you wanted to know, is a directional change in global climate that lasts 3 decades or more. There's a lot of research out there. I encourage you to read and then see what it's doing to the other species that are around us that we either directly interact with or indirectly interact with. So here's a picture of an iguana. It may not even be an iguana. It's just a lizard of some sort. It's a very pretty lizard. I like the turquoise blue, like under the belly area. But what's really sad about this guy is that he's being threatened by global warming. So he may be put on an endangered species list. It could be a she. I don't know. I just default to that pronoun. I apologize if that is offensive. Okay, so now we're almost done. So we're back to this, the core theme of everything, evolution. And one thing that I did not say that I'm going to say now is that the second bullet point, evolution explains patterns of unity and diversity in living organisms. So all of the differences that we see around us today are a result of evolution. And that is what I just said. Okay, so when we talk about all of the different possibilities as far as living things go on the planet Earth, we need to classify the things that we know about so that we have information about them. Cataloging is like, I guess it's just some innate need that we as humans have. It's a smart thing to do though, because it can essentially help you live longer if you know more about the things that are living with you on the planet. So what we've done is we have started to have this hierarchy of, I guess, like categorizing organisms. And so the first thing that we do when we look at these different categories that we're going to stick living things into, we check out the cell. So when we look at the cells of something that is living, we want to know, does it have a nucleus or does it not have a nucleus? And so after we've established that, then we would go further. We would say, okay, well, if it does have a nucleus, is it a plant? Is it an animal? Is it a fungus? Or is it like some single-celled protist that lives in pond water? And then from there, we further break things down. We say maybe like, is it an invertebrate or a vertebrate? In other words, does it have a backbone or not have a backbone? And we just keep kind of like scaling things back until everything fits in a nice, neat category. The issue with the past systems that we had in place to classify living organisms was that we would look strictly at their appearance, so like their physical features. And then the other thing is maybe we would look at like anatomical structure, so like their body, how it was put together. Do they both kind of like, do the things look like each other? Was just the main thing that we were concerned with. And then we realized that there's an even better indicator to tell us how closely related two things actually are. And so once DNA sequencing became a thing, that's what we put into use. We started to look at DNA patterns, and then some of the classification that we had done needed to be redone. Currently, this is still happening. It's an like an ever-going thing, and I'm sure that like with the advent of more tech, there'll probably be some other system that we use to refine things even further. So in the book, they speak to you about, or they don't speak to you, they write to you about domains, kingdom, phylum, class, order, family, genus, species. So what I want to do, because I have space on this slide really quickly, I'm going to list. So I do it in black ink. So this is how we categorize, or this is the levels of categorization that we go through. So first we look at domain. Then we check out what kingdom the organism belongs to. So it's domain, kingdom. And then we look at something called phylum. Phylum, kingdom, phylum, class, order, family, genus, and species. So one word, or not one word, but one thing that I would like to point out, when we were looking at those mice in the very beginning, they had like a really weird naming. It was like, they both began with a P. They were kind of like italicized. Those were their genus and species names. So I didn't want to cross them out. I wanted to highlight them. That was whack. Okay. Well, that'll be memorable because I crossed them out. So I'm going to keep talking. So genus and species is how one scientist may speak to another about an organism, and there's a universal understanding about specifically what organism you're talking about when we do genus and species names. And that is convenient, again, because we need to have like the same language when we're speaking to each other. Otherwise, someone may be talking about a dog and another person could be talking about a wolf. And those are two very different things that you're dealing with, or organisms that you're dealing with. So domain specifically is looking at whether or not something is a prokaryote or a eukaryote. Does it have a nucleus or does it not have a nucleus? Kingdoms, you start to look at whether or not things are plants, animals, are they fungus? And so every time we go further down this path that I just drew, we're getting more and more specific. So do you guys remember what that's called? You should. It's called reductionism. So we're doing the same thing again, but we're not looking at the entire planet with like non-living things included. We're only looking at the living things. All right, so here's the eukaryote domain. So we are eukaryotes. We are animals that have a nucleus in the center of our cells that hold our DNA. We have organelles that do work for our cells, and that's basically it. We belong to this particular domain. We're multicellular, meaning we're made of a bunch of cells. There's some other things that belong to our kingdom as well, fungi and plant. They also belong in our domain, in our kingdom. So here's a picture of these domains. So you got the bacteria domain, you have the archaea domain. Archaea are like ancient bacteria, basically. They are able to do some really, really cool things. They live in places that most other living things could not survive. I've heard them referred to as extremophiles, like they love extreme environments. They're cool. So very interesting. Eukarya is us, so eukaryotes. And then here you go with the kingdom. So plant kingdom, fungi kingdom, animal kingdom, protist kingdom. All right. So what is this slide about? Unity in the diversity of life. So even though we're all really, really different, there are some similarities that can be drawn between different species. And there's this one scientist who you may have heard of. He's kind of a big deal, but you know, I'm not bragging on him or anything. His name is Darwin, and Darwin is the one who came up with this idea of natural selection that you might hear people talk about so fondly. So Darwin went on some expedition on this ship called the HMS Beagle, and he's a really interesting kind of a character, but basically he just did a bunch of observing. He went from island to island in one specific place called the Galapagos, and that's where he found some really huge variation within just all of the animals that he was coming across. But most famously, Darwin, where he documented like these finches, and a finch is a bird if you didn't know, but I'll show you. So after he came back from his travels, he did go on to publish this book called The Origin of Species. It was not popular when it first came out, but like so many other things, once he was well on his way to the grave, there was like a spark of like curiosity to go back and revisit his work, and of course it's like the best thing ever now. So here's a picture of 3 different birds. So you see a flamingo, a robin, and a penguin. Well, they're all birds, but look how different they are. The variation is crazy. So how do we put these guys into specific classes? So Darwin starts to think about how can he kind of like catalog or mark these differences, and then more specifically, what is it that is causing them to be so very different? I mean, they're all birds, right? But when we look at the finches, this is what we see, or what he noted. He noted that generally they're built to be able to sustain themselves in their environment. And if they have favorable traits, then they're able to pass them on to their offspring so they can reproduce and survive. And then you get this competition within the species itself because of the variation in traits. And competition is not necessarily something that is done purposefully, like how we set up a race, or maybe, you know, how we play basketball against each other, something like that. But in this case, there's competition in a sense of like, can you get a mate? Are you able to run fast enough from a predator? Can you spot the prey better than another, you know, organism of the same species? And inevitably, somebody has to lose, or something has to lose. So Darwin, he makes these inferences, and he says, okay, well, if an organism is best suited to its environment, then it's more likely to be able to find a mate, and it's more likely to be able to have babies. And the more successful those babies are, the more and more generations of this trait that's so great will be passed on. So over time, you end up with this shift in the look of a population. And what's interesting about the Galapagos Islands is they're so close to each other, but they are separated by water. And this separation, he believed, caused these finches to develop different features over, you know, maybe not a really, really long period of time, but a nice chunk of time. So, and that's why we call it natural selection. Nature is the one that is pushing this forward. So the temperature, the environment in which an organism lives is going to dictate whether or not that organism is successful. All right, so here's an example of that. You have this population of some beetles. You'll notice that they have different colors. The ones that are darker blend in better with the background. The lighter ones are easier for a predator to see, and so they get eaten. And if they keep being eaten, they cannot reproduce. And then over time, what you'll see is the population will shift. So there will be less and less of these light colored beetles and more of the dark colored beetles. All right, so what we do now that we have this information that we've kind of like laid the groundwork for, that we can classify animals into different let's just say different sections for now, because I'm not able to pull a better word. So we are able to classify these animals, plants, whatever, into different sections. And then we notice that environmental factors can kind of sway the way that the population looks. So we have this natural selection thing at play. At what point does something become like a whole new species? Well, we have to go back first and try to find what the origin of that particular grouping of animals or plants or whatever, what it is. So why do you need the origin point? Well, so that you can see the level of diversity that has happened. And then maybe from there, you can look at whether or not these animals are still able to interact and reproduce successfully. Once an animal can no longer reproduce with something that, I don't know, a couple 100,000 years ago was of the same species, then you've created a new species. Like it's brand new. There's something different about it could be the number of chromosomes. It could be, I don't know, just like the overall DNA patterns don't match up, and so they can't reproduce with each other anymore. So this particular slide is just saying that there's some different things that we're looking at. You have a common structural architecture, then, and maybe you're coming from a common ancestor. Natural selection over millions of years is going to help create some diversity within this tree of life that we speak of. So this is what Darwin says. He says the finch species of the Galapagos Islands are descended from a common ancestor. So why did he think that? Well, if he looked at, not if he, but he did look at the beak shape of these finches. So he's saying there's some ancestral finch, and we may not know exactly what that finch looks like, but we could kind of like, I don't know, we could guess, for lack of a better word. And then what happens is we kind of start to draw out what the current population looks like. All right, so you'll notice that you have like this line extending from the ancestral finch, and then you start doing this thing where you like branch off, and they call those cladograms. So in a cladogram, you're seeing places where a new characteristic popped up. Every time you do a branching, so like I'm going to switch colors to black, like this branching here means that those particular finches, they split from each other. There was some significant change in their physical appearance, in this case, their beak shape, that sparked or spawned a brand new species. So you'll notice here, this is really a good callback actually. So you have like, you see the slanted or italicized names, and they're hard to pronounce. Certhida, I don't even know, Olivacea, I guess. That one is the insect eater. But then if you look at the one underneath of it, it's called the Platyspeza. I don't know how to pronounce the last one. I'm not even going to try. So each of these are the genus and species names. And what you should see is that they actually are now all different species. These guys can no longer mate with each other. And what is responsible for that is the question. And again, I've already given the answer. It's the fact that they have been closed off in different environments. And so over time, whoever had like the best ability to survive, they kind of like had a population explosion. So the insect eaters, wherever they were closed off from, there was a bunch of insects, and if they could get to them, then they could eat and they could survive. And then you just go straight down the list. And again, this branching is just showing you every time you do separate them, there's some brand new feature that pops up. All right, what do I want to say about this? Nothing really. This is just about the scientific process. So this is definition stuff. You guys already know you have to explore and discover, so we don't need to really do too much with that. And in exploring and discovering things, you may gather some evidence. So there's a couple different types of data / evidence that you can gather. You can get qualitative information or qualitative data, and then you can get quantitative. Qualitative has quality. So those are the things that maybe you write what you see. They are not necessarily measurable. They're just what you saw. But quantitative has like the sound of quantity. And if I ask you for a quantity, then you're going to tell me a number. So this particular type of data is going to be some measurement. So here is an example of a famous study. Jane Goodall did this chimpanzee behavior study. She was like on Jimmy Kimmel or something not too long ago. Anyway, I don't know, that was random. But so she collected a bunch of qualitative data. Look at her journal. She writes a bunch of words. She draws some pictures. That's her data. So moving from that, now let's talk about inductive reasoning. So this is when you are making a conclusion based on like what you see. So it's saying here that you can make a generalization from a large number of observations. So they give you an example. All organisms are made of cells. That was based on 2 centuries of microscopic observations. So over the years, anybody who had any type of rudimentary microscope that they made, they kept putting stuff that was living under it, and they kept seeing, oh, cells, cells, cells. So they make all these observations, and then they come up with something that's a very blanketed statement. That's induction. All right. From there, the book is now talking about forming and testing hypotheses. So what is a hypothesis? It's just a rational accounting for a set of observations. It is usually driven by inductive reasoning. And then they give you a definition for an experiment. It's a scientific test. Let's see what else. So how do you form and test your hypothesis? All right here. There's some observation made. You throw a question out there. You come up with a reasonable answer for that question. And the important thing here is that a hypothesis has to be testable. If it's not testable, then throw it out. It's not good. Deductive reasoning kind of goes the other way. So this one is taking information from a general premise and then you make some prediction about it. So you would make a statement first and then test it and see if it's true, versus you observing and then making a statement to see if it's true. So they're backwards each other. So a hypothesis can never be conclusively proven to be true because we can never test every single alternative that there is. All right, we have already done this. So I'm going to just do a callback to our first week when we spoke specifically about this lady drinking tea. And we made a big deal out of it. Like, we really, really stretched that situation as far as we could possibly take it. But you guys came up with the hypothesis. And then at the very end, or like towards the end, we started to discuss a null hypothesis. So the null negates what you believe to be unbelievable. So it's more logical. So your null hypothesis for that one was that this woman could not tell the difference between what order milk and tea was poured in her cup. And you had to state an alternative hypothesis, which was that she could tell the difference. So something as simple as that can be turned into a very big deal. And we ran it through a lot of statistical testing, some computer generated, some human generated trials, and you really didn't come up with a conclusive answer. All we did was either accept or reject our null hypothesis. So here's what is important to remember. The second bullet point: hypothesis can gain credibility by surviving multiple attempts at falsification. And it seems so like cynical, but this is the way that we have to approach things. So we have to look at things kind of like with the side eye. And you can't be so quick to just say, yes, absolutely, that's true. What we can do is we can try to discredit or falsify. Here it is, attempts at falsification. Whatever it is that we are checking out, whatever we're testing. So alternative hypotheses, they are usually eliminated by testing. And in our case, we couldn't eliminate the alternative. We actually had to reject our null. And that was interesting. Okay, so when you can't answer a question addressed by science, then you're not doing anything experimental and you're not truly stating a hypothesis. Like, so you have to remember that the hypotheses that you make have to be testable. They also must be falsifiable. All right, last thing, and then I'm done here. So we're used to science being done in a very linear fashion. Like, every class you've probably taken has always told you, you make a prediction, you create a hypothesis, you do some background research, and then, you go on, you write a procedure, you get materials together, you do the procedure, you collect the data, and then you do it more and more times, like more trials, and then, so on and so forth. So there's this very like point A to point Z kind of a deal for science as it has been presented in the past. But in actuality, there's not a lot of science done that way. Science is more cyclical. So if you look at this particular graphic, it is showing you how each of these items or each of these processes can be connected, and it also shows you the back and forth nature of a lot of these different parts. So each of the next slides, these ones go over specific things that are happening during each of the phases in that diagram before. So forming and testing hypothesis, you can look at that. Notice that this itself is cyclical. You could be stuck in this stage for a very long time. Exploration and discovery. You could be stuck here for a very long time as well. But this would go hand in hand, or it would connect with the hypothesis testing that we were just looking at before. All right, societal benefits and outcomes. Now we're talking about being social with the science, sharing what we see, what we come up with. And then this is like adding to that whole social aspect that I just talked about. So now you have the community is also checking out your claims. Maybe another scientist picks it up, or maybe they peer review your experiment, that kind of a thing. Again, you can be stuck in this cycle for quite some time. All right, so in this particular chapter, we started off with talking about these mice, and now the book is going to attempt to show you how we would use some of the things that we just spoke about to come up with a valid test for these mice, and link it back to the whole idea of diversity within a species and maybe evolution. So let's go forward. So here are the two mice again from the very beginning. Notice that they both are from Florida. There's a map there that shows you that. They both are of the same species, meaning they can mate and they can make offspring. The places that they live are not very far from each other, and there is nothing stopping them from going from the beach to the inland or from the inland to the beach. However, they may not be very successful with getting to that destination because of their fur color. So what a person may say about why it is that the beach mice have a lighter fur color than the inland mice, just from observation. So we could do inductive reasoning where you will watch for a while and then you can kind of make an overall blanketed statement about what's happening, or we can do a deductive thing where we say, okay, well, it's obvious that the predators that are flying above and eating these guys can see them if they leave their particular environment where they're safe. And so we make a prediction. Mice with coloration that does not match the habitat should suffer heavier predation than the native well-matched mice. And in this particular study, the book is saying, or the authors of the book are saying, people actually carried the study out. So here's what they found. Notice first that they had control groups and they had experimental groups. So the controls are the ones who, in effect, like they're not really messing with them. They just kind of monitor them. So here's the control, camouflage control for the light beach habitat. And here's your camouflage control for the inland browner habitat. And then they have these experimental mice. So now these particular ones are going to go, they're going to be moved from their environment into the other. And they're showing you the percentage of attacks that happen on these mice in the two habitats. And I would say that this supports the idea that the fur color is something that is helping them to survive in their specific environment. And if you take them out and put them in a different background, they're easier to spot, therefore more of them are preyed upon. But then the researchers do something else. So yeah, this is what we were just talking about. Controlled experiment, you have a control group, you have an experimental group. They're manipulating factors they call these variables the parts that they are manipulating. So the independent variable is what's manipulated. Dependent is what you are measuring. So in this case, it's the amount of predation that they were looking at. This is talking about how a control can help like weed out an unwanted variable from being tested. However, it doesn't totally eliminate them. This is about theories. Please take a second to read it. I do know that this was in chapter 1. Chapter 1. Hold on, I'm getting a text. Sorry. I will answer her later. Okay, so yeah, this was in the chapter 1 thing. The theory stuff was. My bad. Okay, so now we're looking at something a little bit different. We're looking at the we're looking at two variables essentially. This is what I see. So we're looking at whether or not there is moonlight or if there's no moonlight. And then on top of that, we are looking at the mice being in light colored soil and the mice being in dark colored soil. So there are a couple of things that you should be able to extrapolate from this graph or pull from this graph. I would feel safe saying that in a light colored environment, where there is full moon, so the moon is completely out, there isn't really much difference in the number of mice that are caught based on their fur color. But when there is no moon out, so when it's dark, then it definitely matters. And what's interesting is that when there's no moon out and they're in the light colored soil, the higher predation is happening to the ones with the darker coat. And when I think about that, it sounds very strange because it's like, okay, if it's dark out, why would the darker mice fur not blend in just as well as the light mice fur? I don't know. That's my own question, I guess. And then for this one, you got the number of mice caught in the dark colored environment. It doesn't matter really that much whether or not there's a full moon or no moon. Those white mice are being caught at the same rate. I guess that does make sense because it doesn't matter if it's light or dark as far as the moon being out. The white mice are easier to spot on a black background or a brown background. And if I interpreted this one incorrectly, I feel, confident with talking with a student or two about it and what they think about it. Or nah, it doesn't matter. Alright, that's it. This is the very last slide. So this is just taking everything that we talked about in chapter 1. So the first thing you see, remember the eukaryote and the prokaryote. Next thing is this idea of DNA and passing on genes. You have this energy flow, so energy cycles through an ecosystem, interactions happen, and then you have evolution. So there are your 5 unifying themes, and I am out.